Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. One of the big themes in EV research that's been quite hot in recent years is studying EVs in tissues. So this is something that is difficult. It has its own challenges. And many of you have probably tried to separate EVs from solid tissues after harvesting them from a model or from some patient samples. But we're going to hear about a different approach today, a slightly different approach where you could actually do some things with live animals. And so I'd like to welcome Shannon Macauley to EV Club today. She's at the University of Kentucky now, and she's going to be telling us about this method that she and her group have developed for collecting interstitial fluid EVs from the brain. So Shannon, thank you so much for being on today.
Thank you so much for inviting me to talk about our work. I'm really excited to get feedback from you all, since you guys are all the experts on EVs. What we ended up doing is, I'm an Alzheimer's researcher by trade. I actually have studied for years and years lysosomal storage diseases. And so I have a deep interest in lysosomal biology, especially as it pertains to Alzheimer's disease. And we also have a lot of expertise in my lab for using microdialysis. And so, just to measure things in an awake, freely moving animal as they're doing things. So we can challenge them pharmacologically, behaviorally, and we can actually get at how the brain, this dynamic pool, is changing in real time. And so, about 7 years ago now, we were talking with folks at Wake Forest and about like a grant looking at EVs as biomarkers, and we really started to wonder, what if we could use microdialysis to isolate extracellular vesicles from the brains of live mice? And that's what I'm really going to talk to you about today. And so I just want to start with my acknowledgments, because I can be chatty and I don't want to not thank the people that actually did all this work. So again, this really came out of work from my graduate student, Morgan Pate, who just defended. While I was at Wake Forest, I just moved my lab to the University of Kentucky, but she had a background in lysosomal biology, as did I. And we're talking with Gagan Deep, who's an expert in EVs and exosomes. And we've learned so much from collaborating with him. And we just really had this what if, this wondering question: what would happen? What would we see if we could collect these extracellular vesicles from the hippocampus of live mice? And what would be similar or different from what we see in the world today? And so everything I'm going to talk about is all from her. So I'm a talking head for her work and all the effort that she would put into the development of this. And again, on the exosome side, it's Dr. Deep's lab and his vast expertise in a lot of this. So, I'm going to talk a lot about Alzheimer's today, so I thought I just might start with some facts and figures. So Alzheimer's disease, as we know, is an age-related disease where people suffer from dementia and cognitive impairment. And currently, it's the 6th leading cause of death in the United States, and it's estimated that every minute somebody's diagnosed with Alzheimer's. And while right about now, there's about 6 million individuals living with Alzheimer's disease, with some FDA approved therapeutics, but again, a lot of controversy there. It's estimated that in the next 20 years, 30 years, this is going to get up to about 14 million. So this is a really big public health crisis that we need to figure out, not only how to treat the disease, but how we can stage the disease. And so unlike other age-related diseases like cancer or cardiovascular disease, where there's actually a decrease in deaths associated with them, the deaths associated with Alzheimer's disease continue to climb. I think it's also really important, especially for this talk, but also globally, that you're aware that Alzheimer's disease doesn't hit individuals equally. So 2/3 of women actually get diagnosed with Alzheimer's disease, which is very interesting. And then underrepresented minorities are at an increased risk for Alzheimer's disease. And so this is not only a public health concern, but it's a health disparities concern as well. And that's something that we need to address, especially in Alzheimer's. And then there's this big piece of Alzheimer's disease of the healthcare burden, where it's just, a lot of people lose wages. There's a lot of costs associated with specialized care for people with AD. So what can happen if we can come up with ways to either diagnose the disease earlier, understand the different stages of disease better, or even intervene, right? Our gold standard for all of this. So I think it's worth saying that not all forms of dementia are Alzheimer's disease. This is the most common form of dementia, and it's estimated up until 80% of all forms of dementia are of an Alzheimer's type. There's lots of other forms of dementia that are really guided by different pathologies. And I think it's worth noting that I think this cartoon might be a little misleading because almost no form of dementia is also pure. And so Alzheimer's disease also has vascular dementia components, Lewy bodies, alpha-synuclein deposits, and things like this. And so when you're thinking about this disease, it's always good to remember that one, Alzheimer's disease does not equal dementia, that there's a very specific pathological signature of Alzheimer's disease, but also that when somebody's diagnosed with Alzheimer's disease, they more than likely have pathologies from other terms of neurodegenerative diseases present in their brain. And so what is Alzheimer's disease and how do we define it? So about, maybe about 7 years ago, 10 years ago now, there was a white paper that came out that was a biomarker first definition of AD. And this is what we, have come to be known as the ATN framework, where A stands for amyloid, T stands for tau, and then parenthetically, N stands for neurodegeneration. And the idea was for a long time, Alzheimer's disease was equated with dementia. And what we found from a lot of failed clinical trials, that there's a lot of reasons people get dementia that has nothing to do with Alzheimer's pathology. And so one of the things that I've always been struck by, and in this great review by Rick Vanderkant, is that there's a huge, long preclinical stage with amyloid pathology. So a lot of people have amyloid as you age in your brain, and this does not mean you have Alzheimer's disease. But it's one of these things that's kind of like the canary in the coal mine as an early biomarker of these changes. And one of the things also to understand about Alzheimer's disease in this cartoon of the brain is it kind of starts from the outside in and then spreads with the disease. And so not all regions of the brain get amyloid. They don't get amyloid at the same time. And so you have both this temporal and spatial distribution of amyloid that's kind of a first phase of this disease. Now, tau is different. It kind of has a different spatial distribution and a different temporal distribution. Tau comes a little bit on board a little bit later in this disease, and I think of it as kind of starting from the belly of the brain and spreading out. And so you have actually, and you need both pathologies actually to have a diagnosis of Alzheimer's disease. And it's interaction of these pathologies that give rise to these classic ideas of, cortical shrinkage, brain atrophy, enlargement of the ventricles, and then when people present with cognitive decline. So you have this spatial paradox of this disease. And then you also have what happens with tau is that there's actually age-related changes in tau burden within the brain that have nothing to do with Alzheimer's disease. And so there's kind of this convergence of pathology that really gives rise to AD. And one of the things that we know very well about Alzheimer's disease is that it's more than amyloid and tau. And so, amyloid is just the extracellular accumulation of this beta amyloid protein and a whole host of other things actually within these amyloid plaques. And then neurofibrillary tangles is the intracellular accumulation of the hyperphosphorylated tau protein that causes microtubule destabilization. And it's kind of the interaction of these with all of the other cell types within the brain that give rise to AD. And so what you have fundamentally in a cell autonomous way is these disruptions in proteostasis, but in a cooperative way, alterations in metabolic function, lipid metabolism, oxidative stress. You see this profound neuroinflammatory response that involves not only the glia, the astrocytes and the microglia, but also recruiting cells from the periphery. And then you have this actually also associated with vascular damage and blood-brain barrier breakdown. So this disease is much more than just neurons dying. And there's a lot of really complex mechanisms at play that come on board and off board during the AD continuum that maybe we can, one, use as biomarkers to stage the disease, but also might be telling us about what's happening within the brain. And so one thing that I always like to, kind of highlight is that all these pathological changes start well before somebody ever comes in and complains of Alzheimer's complaints of mild cognitive impairment. So this vertical red dotted line is like symptom onset. When people show up at their neurologist or their PCP and say that they have cognitive issues. And the pathology that's happening, the changes in amyloid, tau, metabolism, are happening 10, 20 years prior to this moment. And a lot of the changes that we see, like changes in amyloid, actually peak and plateau with the onset of clinical symptoms. And so when you're thinking about changes in amyloid, tau, or other biomarkers, it's really interesting to consider when these changes are happening and how can we go earlier and leverage some of these changes as a biomarker for this disease, and what is it telling us about where a patient may be across the AD continuum. So there's a lot of classic biomarkers that have been out there to hammer through this ATN network. Traditionally, we've used imaging biomarkers to look at amyloid and tau. We use FDG PET to look at the hypometabolism associated with neuronal loss, or MRI with atrophy. We can use the CSF to look at changes in the A-beta and tau levels. So for example, with amyloid, this is going to come up today, is that when you have a lot of plaque buildup in the brain, it traps all the A-beta. And so you actually see drops in A-beta 40 in the CSF, or the 42-to-40 ratio. And this is actually indicative of that pathological burden being stuck in the brain. You see increases in total tau and phospho-tau within the CSF, and then other markers like NFL, which has gained a lot of attention within the CSF. There's a lot. This is an outdated slide. The plasma biomarker field is just absolutely amazing right now in the AD space, with changes in being able to, assay amyloid, tau, and other glial and neurodegenerative markers. And I'll save that discussion for another day. But I think what you can start to appreciate about Alzheimer's disease is that this is a very complex disease, and that there's a lot of things going on in response to these proteinopathies that we can then leverage to either stage the disease or intervene. So I think the big thing that comes down to is we have a lot of different mechanisms at play, right? A lot of this happens early on in disease. We have a lot of changes in synaptic and neuronal function. We have changes in amyloid plaques and microglial activation. And then, of course, the formation of neurofibrillary tangles. And so in order for these pathologies to actually happen, it's actually the cooperation of a lot of different cell types. And under normal healthy conditions, they like to communicate via small EVs or in exosomes. And what we know is that in a pro-inflammatory state, which accompanies Alzheimer's disease, whether it's the accumulation of plaques or tangles, you actually see an even bigger increase in some of these EV population. And so what we're really interested in is understanding how this EV population relates to something as simple as a beta pathology and a simple mouse model of Alzheimer's disease. And so I don't think that this slide really needs any introduction to the folks on this call, but there's a lot of different types of extracellular vesicles, right? So we have our apoptotic bodies, which are larger and normally suggesting that the cell's dying. We have microvesicles and which are smaller larger vesicles that actually come off of of blebbing of the plasma membrane. And then we have these really cool little buddies, the small extracellular vesicles, or exosomes, and these are derived from the endosomal lysosomal pathway. And we were really interested, Morgan and I, about these guys because we have an interest in lysosomal dysfunction in Alzheimer's disease and other lysosomal diseases with lysosomal dysfunction. And it was really interesting to us that you had these intraluminal vesicles that were packaged properly, shuttled to the multivesicular bodies, and then they kind of had a fate choice, right? They could be released as exosomes, or they could be going to the lysosome for degradation. And a lot of our initial questions were just really about this switch, and we needed a way to measure this switch. And this is really where we bore out the approach of the in vivo microdialysis. But one of the things that we're also trying to understand is some of the hurdles, that whether the extracellular vesicles could be an attractive biomarker. One thing that we really liked about them is that the EVs are unique to their cell of origin. And so if we had a way of actually selecting for a neuronal astrocyte microglial derived EV, what could that little package of proteins tell us, or metabolites or lipids tell us about microRNAs? about what was happening within the brain. And then this idea that could be protected as these EVs move throughout the circulation was just really exciting. Obviously it's really hard to sample the brain. And with the advent of all these great plasma biomarkers in AD, we were on the bandwagon of, okay, wouldn't it be really cool if we could see what these EVs in the plasma are telling us, but who do we chase and and why? We know that these are good for cell-to-cell communication. So if they're communicating good things, the health of the brain, or bad things like a pro-inflammatory state, are they a way that it's a clearance mechanism for some of these protein aggregates, or just a great biomarker of what the pathology is within the brain? And so we also realized that the field currently had this hurdle, right? Is that these are great because they can be transported across the blood-brain barrier into the CSF or the blood, where they can actively be sampled. But one of the things that we know as AD researchers is that proteostasis is disrupted early in AD, and one of the hallmarks of Alzheimer's is impaired clearance. That means even the smallest proteins and metabolites aren't getting shuttled across the blood-brain barrier into the plasma or into the CSF for clearance. And so if all of these these clearance mechanisms are disrupted, how are EVs selectively getting shuttled and not getting stuck within the brain? And this led us to the next thought of, well, potentially what we're sampling in the brain or the blood isn't specific to the regions that are vulnerable in disease. I showed you a couple minutes ago that we had, that A beta starts in different regions than where tau starts. It doesn't involve the whole brain. There's a lot of regions that are shared. So the EV population that we can sample from the CSF or blood, is this really from those pathological regions? And then, the other thing that comes up is that I would expect, depending upon the state and the progression of the disease, whether amyloid's on board, tau's on board, neurodegeneration, that these EVs actually might have a different composite signature because they're representing different states of the disease. And if we looked at different stages of disease in something as simple as a mouse model, could we actually understand this process a little bit better? And we also had concerns about, postmortem, the postmortem interval, where one of the key things in my lab is understanding metabolism in Alzheimer's disease. And we know that, with our mice, if you sack them and you can't get a brain out within 30 seconds, and then you do metabolomics, that you see a big change in that. So when we think about these human studies where, there's no other way around it with long postmortem intervals, what are we seeing that might not be real, or what are we missing? And then, with the fact that we know that there's impairments in the endosomal lysosomal pathway, are they trapping these either intracellularly or extracellularly? And can we start to get at some of that really fun biology? So I think I've, kind of said, that, there's the traditional approaches have given us so many valuable insights, but there are a couple caveats. One, the postmortem interval, whether the EVs are extracellular or intracellular, and then also sampling distally to the brain, whether or not these EVs that are really unique and signaling at high abundance are actually found in those compartments. And so what we decided to do is leverage our vast experience with in vivo microdialysis to see if we could just isolate EVs from the brain. And the reason we like this is that, one, we can put a probe in wherever we want. So the whole goal originally was to actually put a probe in the hippocampus, which is a region that's prone to Alzheimer's disease, and one into the cerebellum to see how those differ. We realized that quite quickly that it's really easy to put two probes in the brain, but this was a very complicated way to do it. And maybe we should focus on one. That was a bad PI moment on my part. But the other thing is that when we do this, that these animals they're alive, they're unanesthetized, and they're unrestrained. So you put all this stuff in their brain, you insert a probe into the region of interest, and then the mice are just walking around a cage for however you want to sample. And we've sampled up to a week fairly consistently before a probe fails. You can tag a region that's vulnerable to disease or refractory to disease and see how the EV population or proteomics, metabolites change there. It also gives us this unbiased, like heterogeneous pool of EVs, right? So there's some absolutely fantastic work coming out looking at, using human iPSCs and other ways in vitro, but they're pretty selective for different cell types or don't represent the total complexity of the brain. So this is really cool because you're doing it in the brain's native environment. So it might give you a little bit different information that you would see otherwise. And then we also, again, I've said this a million times, that proteostasis is dysregulated in AD, and we just don't know if this pool of EVs is, one, getting secreted from the cell, let alone getting to the CSF, blood, or other compartments. And so, again, we've used microdialysis for a lot of different reasons on a lot of different projects in my lab. And the really interesting thing about this is that it's real time, it's dynamic. You can get other proteins, metabolites, lipids out of that extracellular pool. This is not the CSF, I should say that this is actually in the hippocampus. So where the parenchymal, all the cells are signaling back and forth. And we know from a variety of other studies that this is a highly dynamic pool, and it really is involved in the cooperation of these cells. But what we have the ability to do is not only like isolate and look at EVs, but we can look at those EVs relative to other proteins. And in our studies, we did this relative to that highly dynamic pool of A beta. So just to get on to it, the approach we use actually in retrospect is something simple. Microdialysis can be a bear to set up, but the thing that we just did was found ways to collect longer. So what you end up doing is you infuse ACSF with a little BSA that facilitates the collection of molecules back up into your ISF collection. But you just use this zero flow method. That means whatever you're infusing in, you're taking out. And so you're not changing the absolute concentration of fluid in the brain. And then molecules are just going to freely diffuse apart across your membrane. And then you can collect them in this with a peristaltic pump. And a 4 degrees fraction collector, so it stays super cold once it comes out. And again, what we did is we just collected for four plus days, so that we had enough volume, because you only collect about 60 microliters per hour, so that Dr. Deep's lab could actually isolate EVs from 4 mils of fluid. And the animals walk around, groom, eat, do their normal mousy things, and are relatively unperturbed by the fact they have something, little unicorn thing sticking out of their head. And so the first thing we asked is, we have 2 different ways of doing microdialysis. We have a 38 kilodalton probe, which is for super small metabolites, peptides. We use this to measure A beta. Then we have what we call the mega probe, which is what we use to measure tau, ApoE, other larger species, and extracellular vesicles. And what we thought is, so the 38 kilodalton is actually really easy, a lot more easy to do than this mega probe. And we just wondered, if you compared them, what you would see. We expected that the small kilodalton probe would not collect a lot of things. There shouldn't be a lot of things in there because of their size. And we're actually surprised that we did collect some EVs, and we actually collected more EVs than with the larger probe, but the protein concentration in the larger probe, or in the EVs, was much higher. And if you look at that on a per vesicle number, that we're pretty much capturing empty vesicles. And we're not surprised by this. Unlike other filter systems you'd use in the lab, this is actually a biological membrane, right? So even things that should be bigger can squeeze through smaller holes. It's just they're not packed with anything. So when this showed us, kind of as a control, that the 1000 kilodalton probe, I'll call it the mega probe microdialysis, works at collecting these extracellular vesicles, and use a smaller probe size, you don't get as good protein packed EVs out. The next thing we did, again, in collaboration with Dr. Deep's lab, his lab did all this, was to go head to head with different isolation methods, using ExoQuick. I mean, these studies were initiated about 7 years ago, size exclusion chromatography and ultracentrifugation, just to say, okay, are we getting these guys? What do they look like? And does this change? And what we found is that, kind of interestingly, is that with these different isolation methods, we found that there was a change in, the distribution, the size distribution of the vesicles that we could get out. Interestingly, both of them, it was the ultracentrifugation and the size exclusion chromatography that seemed to get us in that sweet spot of what the small EVs were that we wanted to look at. And it looked like there was kind of a trend that the ultracentrifugation was better at collecting EV concentration. And this was encouraging because, as you can imagine, that our samples are pretty low abundant. So I wanted to maximize the amount of EVs we could get out of them for proteomics and for other things down the road. And so we settled on using ultracentrifugation for our EV isolation approach. And then what we did, or what Dr. Deep's lab did, is actually just investigated whether common EV markers were found on our EV ISF on this dialysate. And what he found is that using immunoglobulin labeling, we saw CD63 and CD9 positive vesicles within our EVs isolated from the ISF. He did FACS labeling that showed that there's CD63 and CD81 positive vesicles as well. So this definitely was, right in there that we're collecting these small EVs with a lot of exosome or EV markers. The next thing we wanted to do was actually look at the proteomics. So we knew from our microdialysis that we were getting a good average particle size, that our concentration was decent, and that we're getting proteins within the cargo of these extracellular vesicles. So what Morgan then went back to do is after we did proteomics with Christina Ferdui's lab, she went and did a bunch of proteomic analysis and pathway analysis using IPA, David, and string. And our first pass was just to understand, what are these guys saying? Is there anything of value here? And what can we learn? And she compared the proteins that we found in our EV ISF with other published or known data sets. And what we found is that we had a lot of overlap between some of these published data sets. We also had some unique proteins, which was, kind of very exciting. If you start to look at these, it looked like that a lot of our unique EV ISF pathways were related to protein or cellular metabolic processes or acute phase response, which again is inflammation, which we found really intriguing. But this actually showed that we are getting EVs that are both unique and similar to what other people have done in very elegant studies before us. We then did some pathway analysis to say, okay, can we confirm that we should, the probe's in the hippocampus, so it should be coming from the hippocampus. And in fact, when you do pathway analysis, it shows that it's hippocampal derived. The bone marrow is really interesting because we have this insane microglial response that I'll talk about in a way. And so I think you're picking up kind of the fact that a lot of microglia have the same markers as macrophages and other bone marrow derived cells. So that was really exciting. And then if you look at cellular components, we actually saw that, we're hitting this extracellular exosome. So we're collecting exactly what we want to be collecting from the region that we want to be collecting. So this was a really nice pathway analysis confirmation of what we thought we were already doing. So then the next thing that Morgan did is said, okay, this is our EV ISF. How does this relate to published data sets that use human brain tissue EVs or mouse brain tissue EVs? And what we found with our proteins, we don't get as many proteins, where we need to work and develop more sensitive methods to get at the number and the quantitative changes of proteins we see. So if anybody wants to collaborate, give me a call. But we do have also a unique set of proteins that are specific to our EVs. And when you start to look at these seem to be, again, picking up a lot of these really interesting like endothelial cell neurovascular coupling, immunometabolic changes, and also, proteostasis and RNA processing. We also have, proteins that are shared that we can see in our EVs that are known to be in human brain tissue EVs. And that was really exciting. And so this was something that, we want to pursue further with human specimens and such. So what we ended up doing is that we were fairly confident that we can get the EVs out, that they have, that they're similar and also different from what other people have published. And so we really wanted to know how they change relative to Alzheimer's disease. And so I'll be the first to admit there's no great disease of Alzheimer's, or sorry, model of Alzheimer's disease. No model recapitulates in total what's happening in AD. It just doesn't exist. But what you can do is, if you understand the caveats of all of your model, you can model aspects of disease. And so the APP PS1 mice is this overexpression. They have these two mutations that make humanized A beta, and you can get one, A beta overexpression and amyloid plaque formation. So this is great because we can model that proteinopathy in this mouse model. This mouse model does not get tau pathology. And mice do not go from, unless you actually put even more genes in where you overexpress both, you do not have what happens in AD, where A beta pathology then begets neurofibrillary tangles. So what's really interesting about this is just a coronal section of the brain at 3 months, even though they're overexpressing A beta. So there's a lot of A beta, the soluble A beta on board. It's not aggregated. And then as you age, as the animals age, beginning at 4 to 6 months of age, you start to see little polka dotted plaques. And then by an super aged animal, 18 months, it looks like, it's just plaques aplenty within the cortex and then the hippocampus. One of the things that's interesting is that the female mice get plaques sooner than the males. This is also, we don't know if this is transgene per se, something else biologically happening, but that's what we see in humans, right? Females get AD at a higher rate than males do. And then there's also other aspects of the pathology that's mimicked in these mice. So this is a micrograph where it's triple labeled with X34, which stains the amyloid plaques. The red is actually microglia. So they surround these plaques and they kind of wall them off. And we actually think that early on, this is a protective mechanisms. The green is astrocytes. They're also there. They're periplaque astrocytes. And then these immune cells, these glial cells, are actually, again, packing down those plaques. So they're less toxic to the neuropil. And so you have not only this overproduction of A beta, you have this protein aggregation, but you also have the glial response to these extracellular plaques that's all mimicked in this model. And so I think, there are some cognitive and behavioral deficits that happen in this model, but just like in Alzheimer's disease, like amyloid is not responsible for mild cognitive impairment. You need tau on board. And we see that with these animals. There's some things that go a little awry, but for the most part, it's the tauopathy in AD that drives the behavioral deficits. So what was our study design? So we had 4 groups. We had 3-month-old, either wild type mice or these APP mice. So this again is before they have any plaques in their brain, but they are overexpressing A beta from birth. We have 9-month wild types. So they're just a middle-aged model mouse. And then we have these 9-month APP mice that actually get plaques to a pretty solid state. And we actually in this study did males and females. And we did our microdialysis, and what we collected was enough fluid for EV isolation. But also what we did is held back a little bit of that interstitial fluid so that we could do ELISAs to look at ISF A beta 40 and 42. And what this allowed us to do with our analyses is see how some of these EV populations track, relative to changes in A beta 40, 42, and then amyloid pathology and other markers that we used on postmortem histological sections. So we did mass spec to look at the proteome, and we did pathway analysis to actually see what pathways are changing. And then we took these proteins one step further, and we used Ben Barris's, the Barris Labs, a database that has gone through and said what genes are specific to different cell types, or found in different cell types. I shouldn't say specific. And what we went through is took every single protein that we uncovered and asked, okay, what cell makes you? And so you can imagine proteins can be made in a variety of different cells, like GAPDH is one that's probably in every cell type because it's important for glycolysis. So then it would be identified as an oligodendrocyte, microglia, endothelial cell, and so on protein. And then there's other proteins like GFAP, which is predominantly an astrocytic protein. And so then that would be, and we went through and labeled all the protein hits we got and asked what cell types they could have come from. What was their cell type of origin? And so this is what we see. So from a pathological standpoint, we had 3-month and 9-month-old animals, and we had males and females. And what we saw is similar to what's been previously shown, is that the females get more plaque pathology. You can see within the cortex here that there's a lot of plaques. Within the hippocampus, we see it as well. And although there is plaque pathology in male mice, it's just not to the same extent that we see in the female mice in either the cortex or the hippocampus. So this is emulating and consistent with what people have published on this model for years. We took this one step further, and we're really thinking about amyloid aggregation and the soluble forms of A beta. So there's multiple forms of A beta, and the A beta 40, 42 just depends upon like the peptide length. But what we know from human studies is, as you get those plaques forming in your brain, that the levels drop in the CSF. So there's drops in CSF A beta 42 that are actually indicative of the aggregatory processes that happen in your brain. And so we asked the question, how does changes in the soluble pool of A beta, again, we're looking at these A beta monomers, not these aggregates, how are they changing relative to age and transgene and sex? And so one thing that's really important for this is Dave Holtzman and John Cerrito have extensively demonstrated that one of the main drivers for A beta reliefs is just synaptic activity. So we're making A beta all the time. It just isn't problematic because we have clearance mechanisms to gobble it up and get it out of that extracellular space. And it only becomes problematic if you produce too much or you don't clear enough. And that's when you start to have these aggregatory events that form amyloid plaques. But what we wanted to know is in our mice, if you look at A beta 40, so 40 is thought to be more of the activity dependent molecule, where 42 is thought to be more of the pro-aggregatory form of A beta 42. And actually in Alzheimer's, we use the ratio of 42 to 40 as actually a biomarker. And so what we found is that the females have, kind of this activity dependent level of A beta is elevated more in the female mice than the male mice, which we thought was interesting. And moreover, you see a drop in the male mice as they age, which you don't see in the female mice. And then what we do see with A beta 42 is that the levels are high when the animals are young and there's no plaques. But once you get those plaques, you see in both the male and the female mice, A beta 40 levels, drop. And so even the species of A beta in this dynamic interstitial pool are doing something differently and relaying different information about, one, neuronal activity, than two, the aggregatory process that could be impacted by or the EV population. So the first thing we asked was to characterize the EV number, the particle number. And what we found rather interestingly is that if you separate the females from the male mice, that there's an age-related increase in the number of EVs in both the wild type and APP mice. And interestingly, the APP mice have less than the aged wild type. But this seems to be really driven by an age-related phenomenon, more so than actually a pathology-dependent phenomenon. And we didn't see this in the female mice, which we find absolutely interesting. If you look at the EV size, we're selecting for size, but they're pretty much, so there's really no big difference in size. I do wonder if we didn't filter for this specific size range, what we would see here. But again, this data is consistent with what we would think given our experimental approach. Then we went ahead and did, some sort of pathway analysis to look at these guys, either as a function of age and wild type or pathology in our APP mice. And one of the things that was absolutely striking to us is that we thought, maybe naively, that when animals would get pathology, that they would have like an upregulation of all of these proteins, and they'd be super unique to like the animals with pathology. And that's just actually not true. So it's actually the young wild type that have the most robust proteome, the most diverse proteome. And when it really comes down to it, a lot of the proteins that are in all of these EVs are shared. And one of the limitations of our studies is that we were able to identify what proteins were there, but we weren't able with our approach to really look at relative changes in a specific protein level. So that is something we're working on. But nevertheless, the wild type had a couple changes. This HMGB1 is actually associated with cognitive decline in Alzheimer's, which I think is interesting that it seems to be specific to an age-related phenotype and not necessarily a beta-related one. But then what we also found is that there's a 5 proteins that we never would have predicted that seem to be specific to the cargo of our APP mice. And so we're exploring whether these could be new potential AB-specific or A-beta-specific biomarkers, I should say. And then we also saw that there was 168 proteins that were shared in all of our samples, and whether this could be a new brain or hippocampal specific signature for the population that we're taking out. So it's very data rich, and we're still trying to tease through what we can gather from all of this, but there's some really neat things that we're uncovering. We then did some pathway analysis to just see how these different pathways were clustered, and we loosely clustered them into these different groups. And what we found is that really when it came down to it, that proteostasis was really highly represented in all our samples, whether you looked at young, old, pathology, not. And RNA processing was also highly abundant. There's also, if you look at the glucose metabolism, immune function, and lipid metabolism, there's this really neat immunometabolic signature that's coming that's highly represented in our EVs, which we thought, again, was very exciting, especially given some of our microglial results. And so we did some intersection plots just to look between, at all these different groups and how complicated it is. And what we found is that, 25% of young wild type animals, their pathways seem to relate to RNA processing. And then it didn't matter if these animals were old with pathology or young with pathology. We saw a decrease in these pathways, suggesting that RNA processing, that these EVs are trying to communicate something about that to one another. The other one that was probably not surprising was this change in proteostasis, is that proteostasis-related pathways were fairly consistently elevated in young animals, old wild-type animals, and young PSAPP animals, but we actually saw a drop in the proteostasis pathways once you started to have amyloid beta aggregation. So this seemed consistent to what we would have hypothesized looking at these diseases. One of the neatest things about this is that we found that the wild type animals and the APP animals actually had an increase in protein concentration compared to the young animals, either with or without the transgene. But what was shocking to us is that you had a decrease in the protein diversity. So what this means is that if you look at the proteins, not their relative abundance, but whether or not it was present or absent from a sample, is that if you had animals that were young with a beta overproduction or aged or with pathology, that you saw this decrease in the accumulation, decrease in the protein diversity within these samples. And I think that was surprising to us because we thought we'd uncover a lot of really new proteins, which we really didn't. So again, age, genotype, and pathology seem to all decrease the protein diversity while increasing the protein concentration. So that means that there's more of kind of the same proteins in these EVs. So they're really trying to signal something pretty interesting. And then when we started to look at pathway analysis and just took more of the top 20 pathways that we're seeing either in young wild type, aged wild type, or APP mice, that a lot of the pathways seems, again, relate to proteostasis. And then as you start to get changes in A beta production and aggregation, that you start to see the inflammation immune cell pathways on board, as well as changes in metabolism. So we think that this ISF EV population has really got a really strong immunometabolic phenotype. So then, as I said earlier, we went and took these pathologies and we actually associate them back with specific cell types. And again, one protein can be represented in all the cell types, or it could be unique to that. And we actually only counted proteins that were found in 2 or more of our samples. And then we actually asked how any of this was related to changes in A beta pathology and stuff like that, going forward. And so what we found, if you look at the endothelial cell proteome, is that as the animals either have pathology or get older, or have, they're overexpress A beta or get pathology, we see a reduction in the endothelial cell derived proteome. And one of the things that I think might be happening here is that, again, we're sampling from that interstitial fluid. I wonder how much of this is being dumped into the blood. And that is not something that we measured. We have the blood and we just haven't gotten there yet. But whether or not the way in which the endothelial cells are going to be signaling could differ depending upon the stage of the disease, and where this might be a great way where a sampling from the blood might be far superior than our way of sampling from the parenchyma if you're interested in vascular related EVs. Interesting, our endothelial cell population didn't change in terms of the percentage of proteins that seem to come from that. Again, this doesn't mean that the types of proteins didn't change, but just the percentage that made up the whole of endothelial cell derived was fairly consistent, one across genotype, age, and sex. The neuronal EV proteome was really interesting. So as I said before, when neurons are just active, they make ISF-A beta. And as long as it's cleared, it's not problematic. But in Alzheimer's disease, those plaques act as a magnet and gobble up all that A beta. And so this results in a decrease in ISF-A beta with an increase of amyloid pathology. And we found something really interesting with our neuronal derived proteome, where we see that actually in aged animals, that there's a decrease in the percentage of proteins that are found in neurons. And now, as an AD researcher, this is not surprising because we know proteostasis is absolutely and protein synthesis is disrupted in this disease. But it was still interesting to know, since a lot of work is being done with neuron-derived exosomes or EVs in this disease. What we found is that the number of EVs from neurons actually correlated very well with ISF-A beta 40. And what we think this is actually reflective of is if you're making A beta 40 and you're clearing it, and this is very dynamic pool, then it's a healthier brain. And it's really, once you start to get those aggregation of A beta, that you see these drops in 40 and 42. And the same relationship holds up with A beta 40. But what we don't see is any relationship between the neuronal derived proteins and A beta aggregation. So this means that it's almost a loss of the function rather than a gain of function here. When you look at astrocytes, you see a similar thing, that there seems to be again, across the board, it's fairly consistent with the number of astrocyte-related proteins that we see. I believe if we had more animals, we'd actually see a really interesting sex difference here. But we see that it's highly correlated to A beta 40 levels in the interstitial fluid. And again, astrocytes and neurons talk a lot in order to fire during synaptic activity. And so we think that this is reflecting a healthy brain, and that these proteins, once they come off board, it's the loss of function, not a gain of function, in terms of how we analyze this data. Now, what I would expect is that if you look on specific proteins, that their levels are going to change, with pathology and demonstrate a new relationship. The most interesting relationships we saw was within the microglial EV proteome. And so what we saw is that if you look at all the genders combined, that the level of this proteome of the number of microglial EV proteins was fairly consistent. But what was really interesting is that there's these really strong relationships with A beta 40 and 42. And so as, again, A beta 40 and 42 drop, which is when it's aggregating in the brain, this is when you see the most EV microglial proteins in the EVs, which means that seems to be in response to this aggregatory event. But we also, again, saw this really interesting sex difference. So we went back to staining our tissue. And again, as I've mentioned before, we have blue can stain the plaques, red can stain the microglia, and green can stain the astrocytes. And in the AD brain, a lot of when you have A beta on board, a lot of the glial activation is periplaque. It's associated with sequestering and compacting that A beta aggregation. And when we looked at CD8 positive microglia, so this is across the whole brain, we actually see that there's more increases in CD positive microglia throughout the brain than we saw with the male mice. And we see that the CD68 has a positive correlation with plaque burden, but only in the male mice. So we decided to refine our analyses a little bit better and look just at plaque-associated microglia. And in the interest of time, so we have time for a talk, I'm just going to breeze through this. But what we found is that if you look at the female mice early on, they start to get plaques earlier. And they also seem to get CD68 earlier, but their microglia are not associated with plaques in the same way that those male the microglia and the male mice are. So it suggests that there's this sexually dimorphic response of the plaque-associated microglia that seems to be related to the EV proteome. And so looking at this a little bit further, what we find is that if you actually look at the females and the proteins that they're secreting within their EV-ISF, is that there's no relationship with the changes in A-beta or tau, A-beta 40 or 42 or plaque pathology, most likely because it doesn't seem like the female microglia are associating with plaques properly, where we see that there's these very strong relationships between the plaque-associated microglia, A-beta 40, 42, and these proteins, but it's specific in the male mice. And one step further, we looked at the age-related, the pathways of those microglial proteins in female and male mice. And what we saw in the female mice is that you have this positive regulation of two Alzheimer's-related pathways for both amyloid plaque formation and as well as tangle formation. And it's driven by two AD risk genes, clusterin and ApoE. So this is really neat that this is kind of uncovering some sexually dimorphic response of the microglia. It seems to be mediated by the EVs, and it might be telling us something really unique and interesting about this disease. So to just summarize, what we did is developed a novel way to isolate the EVs from the interstitial fluid of the hippocampus. We characterized these with Dr. Deep's lab to look at whether they had the markers of a lot of the EV population. And then we did pathway analysis and kind of looked in a cell-specific, air quote, way at how these change relative to age, pathology, and sex, and saw a really interesting sexually dimorphic response with the microglial EV proteome. And so again, I think we'll just stop there so we have time for questions, but big shout out to Morgan, who this was her labor of love. And I mean, I'm just so proud of this paper. It's on Bioarchive, it's under review, hopefully it'll be out soon. And then Dr. Deep Gagan, who's just a fantastic collaborator. And with that, I'll take any questions.
All right. Well, thank you very much, Shannon, for sharing this, and congratulations to you and Morgan and everybody on the team for this exciting work.
Thanks.
We have very limited time for questions today, so I think that I am just going to ask these questions, and if people need to unmute or follow up, please do that. I will also, Shannon, I will share with you the contents of the chat box, so that if you see some questions that we are not able to get to today, because we have quite a few, you can feel free to follow up there. And I would also encourage anybody to contact Dr. Macauley and her team with questions. All right, so let's just start here with Phil Askinase asks a question about what about the RNA. So have you looked at RNA, or do you have any plans to look at RNA?
Yeah, so we haven't done it yet, but Gagan is actually looking at this right now. He's uncovered some really interesting, observations in the human AD biomarkers that he's looking at, but I don't have any results to share with you today.
Okay, great. Yeah, we'll look forward to learning more about that. Then John Moniror asks a question about administering drugs. So if you wanted to study a drug of abuse, he says, would it be possible to modify the system or add a separate cannula to infuse a drug prior to or while you sample the EVs from the ISF?
Yes, 1000%, John. So this is actually what we set out to do. We wanted to, so you can use reverse microdialysis. So there's two ways you can do it. So you can actually go right into the perfusate, so into that ACSF with drugs to modulate whatever you want to do. So we do this with we do a lot with diabetes drugs. You can add sucrose, you can do whatever you want, and that'll reach that site, change that microenvironment. Then you can collect what the changes are in that EV. The goal was to modulate actually lysosomal degradation and exosome biogenesis to see how all of this would interrelate. But again, developing the technique was a bear, but you absolutely can. They also have microdialysis probes that have a separate infusion pump or a port. And we've done things where you've cannulated the jugular and the femoral artery with microdialysis. So lots of opportunity there. Yep.
Yeah, great question and lots of opportunities. Next is a question. This is going back to your methods, I should say. The choice of ultracentrifugation over size exclusion and precipitation. I know you went into some detail about that and the measurements that you did. I was actually a little, personally, I was a little bit surprised at what you found with the precipitation, which was very interesting to me, because you found that with precipitation, you actually had larger particles, whereas typically in other systems, when you have precipitation, you actually get a lot more of the smaller things that are just coming down with that polymer. So I wondered if you could talk a little bit more about that, and maybe mention why you think that there were those larger particles with precipitation.
So the short answer is no. So this was really, where Gagan's expertise. So that data was generated in his lab. So the short answer is I'm not sure. And one of the things that I am interested in is if we don't use, filter out like the specific smaller EV population, what really would we get, right, with larger vesicles and things like that. But to be honest, that's outside of my realm of expertise, but I can certainly follow up with Gagan and get you an answer. So
great. Okay. There's a question here about diagnosis. So what's would it be possible to use this method in humans, or are you rather identifying markers that could be looked for elsewhere?
So there is reasons why people go into the brains and actually can get that interstitial fluid out. I don't think that that's really what we're aiming for here. I think what we're trying to see is, okay, what's being produced locally, and then let's chase it in these distal compartments and see whether these little guys are getting out, or are they just local communicators, and this isn't the best one. So we're looking in the blood where I have some ideas on how to collect CSF and look at the CSF pool versus the ISF in the same animal. And we have blood where we're trying to figure out, and then the goal would be to then go back and compare it to the human plasma biomarkers that are known and so well characterized.
Indeed. Okay. So unfortunately, I am going to have to go because I have a bit of a hard stop here. But one more question from Randy Stout, who asks about the pathway analysis and how you found these cell-cell junction pathways. He says that he sees a large amount of gap junction proteins in EVs, and he was surprised that it was adherence junctions that you saw instead of the gap junction or the tight junction proteins. Can you comment on that, perhaps?
That's a really specific one. I'm happy, if you want to reach out, I'm happy to look back in our data set and see. I think the short answer is I don't know the answer to that. I'd have to look back in our data sets. We weren't specifically focusing on that. So it might have just been the way we presented our data, but I'm actually completely happy to share what proteins we found in different cell types if somebody wants to do their own analysis. So
fantastic. Okay, so Randy, Ellen, Wakar, Tanina, I'm very sorry we won't get to your question here, but I will again share this with Shannon, and please feel free to reach out to her with further questions, and anybody else too who didn't get a chance to put their comment in. So Shannon, I want to thank you so much. I think you've shown us that there are some new horizons here in harvesting brain EVs, and we'll look forward to seeing this paper in print hopefully very soon, and wish you and your team all the best at University of Kentucky and your collaborators. So thanks again for joining us, and thanks everybody for being on the EV Club today. We hope to see you again at a future session.
All right. Thank you.
Take care,
Appreciate
everyone.